and her name is Sofiya Micheva-Viteva, and she's a staff scientist with the lab's Biosecurity and Public Health group. She obtained her bachelor's and master's degrees of science at Sofiya University in microbiology and industrial biotechnology, respectively. She went on to obtain her PhD from Rutgers University, specifically from the Robert Wood Johnson Medical School. She believes strongly that fundamental discoveries in the bioscience field should be applied for public health protection. Among her strong interests is studying the mechanics of RNA plasticity, responsible for gene dosage control that are crucial for bacterial survival. Now, there was other stuff in there, but I knew the words, but I didn't know what they meant. So I picked the part that I could kind of grasp. So please welcome our speaker for tonight, Sofiya.
Thanks for coming. It's a very new environment for me to give a talk, so if you can't hear me on the back, I can yell more. But yes, antibiotic resistance obviously is a hot topic. Everybody is interested. And I see it as a race between a very complex system, humans, and a very simple system, bacteria. And that race started in the mid-20th century. And at some point, we thought we won it. And those were the happy 1980s, when people thought that we have all of the antibacterial drugs in place to take care of infectious disease, particularly caused by bacteria, because viruses, we still can't deal with them. And most of the drugs that we usually take today have been invented during the 1960s. There was a boom, 1670s, and then there is a big group of different antimicrobials classes, and some of them are wild spectrum. They target all kinds of bacteria. Some of them are specialized for particular group called gram-negative, and those are mostly the pathogens that we suffer from, but also gram-positive like streptococci. And what happened is that people thought that antibiotics, they can completely destroy the bacteria. Even during the 60s, there were individual studies showing that bacteria that is responsive to antibiotics still cannot be killed. And since then, they were called persisters. And this is non-genetic trait of the bacteria when they go silent, or we call them dormant, and they respond to no kind of antibiotics. They can be even very resistant to treatment with alcohol, bleach, and this form is now considered one of the unknown forms of antibiotic resistance that even the Department of Defense is sponsoring. Our group is part of a big project that tries to figure out what are the genetic determinants of this type of antibiotic resistance. But mostly resistance is caused by genetic traits. So genetic traits are those that bacteria acquires. It's in the genome, DNA encoded. This could be some mutations that make bacteria non-responsive to drugs, or it acquires genomic material that secretes enzymes that destroy the drug, and it makes it not potent anymore. And the concept of drug resistance started to emerge into the scientific field more into the late 80s, beginning of 90s. But what was even worse, the multi-drug resistance, when bacteria can be resistant to all sorts, up to 5 so far have to be found, types of antibiotics. And usually antibiotics are targeting different genetic traits. So even now it's a practice in the medicine to prescribe only one type of antibiotic. If it doesn't work, then they'll go for another. Rarely cocktails of antibiotics are prescribed just to avoid drug resistance. But so the origins of drug resistance are also debated in the science. Some people think it's because of overusing of antibiotics. I remember still in the 80s as a child, being sick, you go after a viral infection, they always prescribe antibiotics, or even at the beginning of the symptoms of like high temperature, they'll prescribe antibiotics. And also antibiotics are in the agriculture a lot used, and then they, the notion is that whatever bacteria grows on chickens and pigs and all the animals that we consume, we can get them, and they have been exposed already to a broad spectrum of antibiotics. That's why they are not responding to treatment. But people have found their publications in Nature Science finding multi-drug resistant bacteria in caves where there is no human activity recorded. So naturally, bacteria can be resistant to multiple types of antibiotics, even when it's native. It has never seen this type. And it has been seen also in bacteria that's not pathogenic, it's mostly in the soil. Those bacteria, they have very rich genome to use all kinds of sources as food, and they can even metabolize antibiotics as food. And the problem is that when this bacteria, by some chance, acquires pathogenic traits from bacteria that has been expelled from animals and humans, they can recombine and then, or they can pass their traits to pathogenic. So the pathogenic bacteria becomes multi-drug resistance. So whatever the mechanisms of drug resistance, our responsibility as scientists is to find some cure, at least for the future generation. We're still living in times when antibiotics are working. Thanks God, very few cases of multi-drug resistance are registered, but they're throughout the whole world. I have here a map, but I can't show it to all of you. And it shows that antibiotic resistance has been found even in Siberia, in places where you don't find high density of populations or people being overexposed to antibiotic treatments. So what we do in the labs is to find novel targets for developing of drugs. So far, when we say novel class of antibiotics, it means each class targets particular genetic trait. Like tetracyclines, they're targeting the ribosome and production of proteins. So just for the general public, what are the ribosomes? Those are the factories that produce proteins in the cells. Many of the antibiotics are targeting them. Also, antibiotics are targeting the cell wall synthesis of the bacteria, and bacteria counteracts by enzymes that are destroying them. And this is one of the major problems of fighting antibiotic resistance, is not just genetic mutations, but acquiring enzymes that are destroying the antibiotics. So what we are looking for is for antibiotics or compounds. Before they become antibiotics, they're just compound. It's a chemical structure. And we try to find, though first we have to validate a new target. So in our lab, we work on a particular enzyme in bacteria that's important for survival of the bacteria because it keeps the right amount of protein level in bacteria by destroying those that are overabundant. And also it's important for the formation of those factories of protein production, the ribosomes. In bacteria, so ribosomes, it's like ribonucleic acid. In humans and bacteria, not just humans, but eukaryotes, there are different mechanisms of maturation or formation of the ribosomes. In bacteria, it's like one long string is produced, and then this type of enzyme that we're working on is called endoribonuclease because it cuts inside. But it's very selective. so it doesn't cut anywhere, but just particular spot that's important to release the right size of the ribonucleic macromolecules, and then they assume particular conformation, and then they can, like when they come together with proteins, they form a functional factory or ribosome. So if we inhibit this enzyme, the ribosomes cannot be formed, and also there will be accumulation of toxic proteins for the bacterial cells. So it's like one enzyme having broad application for the cell, it's a weak spot. So when you develop antibiotics, you're looking for the weak spot. And also, when you have enzyme that's a target for drug development, you still can find initially something that's effective in inhibiting its function, and then over time it loses effectiveness because enzymes usually, they acquire some mutations and they change binding affinities to those drugs that we have developed initially, so the drug potency is lost. And what we look is to develop drugs that will not be dependent on changes that will cause the enzyme to remain function. For example, there are some changes that don't change the function, but they change binding of the drug. So then drug becomes not effective. The function of enzyme is the same. So the bug acquires antibiotic resistance. So we're looking at particular molecules, not molecules, but so those are macromolecules, and they have small monomeric structures. Like in enzymes, those are amino acids that build the protein. So we're looking at particular important amino acids that are, if they mutate, the enzyme function is destroyed. So we target those for the development of the novel generation of drugs. And how we do that, first we perform a large screening assay. We develop biochemical assay that monitors the activity of the enzyme, and then we add drug after drug and see when this activity is stopped or canceled. And then we start accumulating different drug structures and looking at those structures and the enzyme structure. So how do you know structure? It's a very kind of a long process of figuring out the protein structure is developed by X-rays, and then it's entire science, and we have one of the best scientists in the lab that are solving protein structures. And then we solve them together with the drug, and then we solve them together with the enzyme's target, or so-called substrate. And then we find the right conformations and the right structures of the drug to figure out what is the most conserved structure of the enzyme and the drug that will avoid development of resistance. And so everybody that has been in the field of like developing of new drugs, anti-cancer, antimicrobial, all kinds of drugs for neurological disease, it's very difficult to pass a drug to from the discovery bench to the doctor's office. So there could be like 10 years of stages of testing the drug efficiency, and it's very expensive. So drugs that make money are the ones that the pharmaceutical companies are focusing on. Antibiotics don't make so much money, and that's why our focus right now is to screen drugs that are already approved by FDA, that are in our kitchens also. And not all of them are drugs. Some of them are plant derivatives, which function is not well still known, but they They are, some of them are known antimicrobials, but the function, how they work exactly, is not known. So when we have this plant derivative or drug that has been developed for totally different purpose, then we can find those structures that are the best for inhibition of the bacterial enzyme function, and then we know their toxicity. Some of them are not much toxic. Everything is in the dosage. So they're not toxic when they're used for their particular application, but for example, as antimicrobials, they have to be used like 100 times higher or 10 times higher, because they have to get into the microbial cell, which is more difficult. It has different structure from human cell. That's why we have we have kind of a library of knowledge. We know what's the function of the drug, or the function it has been developed for. We know the pharmacokinetics and toxicity, and then the structure that's important for inhibition, and then starts the whole process of rational design of new drugs. So what's interesting, I just printed a list of our top 10 drugs that showed efficiency in inhibition of the enzyme function, but there is efficiency in biochemical reaction, and then when you test them in bacteria, not all of them are efficient. Those that were efficient in bacteria is what was interesting, something that we have in wine. I just lost my tannic acid. So tannins. So tannins, they have been also, we tested them, and they have effect even in the concentration that usually ampicillin will have. But they are not, they don't kill the cell, they just suppress the function of the, like growth. So even some of the antibiotics currently that are used have mostly suppressive function. When you withdraw them, the cells continue growing. But if they're the only thing that works, doctors may still prescribe them, and then it just depends on the immune system to take care of the cells when they're not multiplying. And this is for the healthy immune system, of course. But so another like strong effect that has is gastric acid secretion inhibitor. But like when we tested in the cell culture to inhibit the growth of the cell, it actually can kill, but in concentration that's like 1,000 higher than the one that we take. To kill, to suppress, it can be 100 times. So how do we improve them? As I said, it's the rational drug design. When we have structures that we know inhibit in biochemical assays, then we know structures that get well into the cell, so it's kind of metamorphosis thing. And still, it's a lot of like math that is applied to make algorithms that can calculate the energy of molecules and how they interact, the energy of interaction, so-called molecular dynamics. So those kind of algorithms are used for the rational drug design. And just to conclude about the types of resistance, antimicrobial resistance, one of the projects that we have sponsored by DITRA, or the Department of Threat Reduction, they sponsor very basic research or very advanced research. So one of these basic research is to figure out what are the mechanisms that make, like molecular mechanisms, if there is any genetic trait that makes bacteria silent or dormant, and then it becomes resistant to all classes of antibiotics because it's not dividing. And then those are so-called persisters. And usually persisters, they're found in all kinds of bacteria. And the one that we usually get sick of are so-called opportunistic pathogens. They're not usually pathogenic until your immune system drops. And then the drugs that are currently prescribed can kill most of them, but then there is a 0.1% of the population that doesn't die because they're dormant, they sleep. And usually the immune system takes care of them, but many diseases like tuberculosis and some exotic disease that start getting transferred in our continent by our troops because they go to Asia, like Burkholderia pseudomalia, it's the causative agent of melioidosis. It's like mycobacteria, like tuberculosis. They are very slow growers, and when they get into the human body, they get surrounded by scar tissue, so antibiotics cannot target them. So usually nobody tries to take care of this kind of bacteria, like dormant